Hi everyone, welcome to Precision NanoSystems Teatime Webinar series. Today we task Dr. Jason Coleman, Clinical Application Scientist Lead at Precision NanoSystems. Welcome, Jason. And he's going to talk about scale-up and manufacturing of RNA-LNPs for COVID-19 vaccine. Before starting, let's have a look at our European team. We have Richard, Suha, and Ed are based in the UK. Jürgen and Martin are based in Germany, and I am based in France. If you want to know more about our platform or technology, please do feel free to reach us out. With that, I appreciate you're here, Jason, and welcome again, and over to you.
All right, hi everybody. I'm very excited to be here with you today. My name is Jason Coleman. Sarah mentioned I'm a clinical application scientist lead with Precision NanoSystems. I'm actually located in the United States outside of Philadelphia. But yeah, it's nice to be here. And really my role with PNI currently as a clinical application scientist is really to help clients, support clients as they scale up their formulations, as they go from the bench scale to larger scale, develop downstream processes, and ultimately get to the clinic and then eventually commercialization. So just to give an overview here, with Precision and basically ultimately kind of what it ultimately takes to develop a self-amplifying RNA-LNP vaccine, or really in that case, an RNA-LNP, genomic medicine in general. We kind of talk a lot of times in terms of a pillar system, or what are the basic requirements to develop the formulation and what you'll need. So just to go over this, so to start, so for a COVID vaccine, we obviously in this case have our disease target. We know we're trying to treat. We're trying to use an RNA that'll replicate the COVID spike protein. So then under this pillar, for a genetic payload, as I mentioned in the title, we're using self-amplifying RNA versus more traditional messenger RNA, because obviously the COVID vaccines on the market today, from Moderna's and BioNTech, they're 100 microgram and 35 microgram dosage forms. But the nice thing with self-amplifying RNA is the ability to use a much lower dosage, because of the self-amplifying characteristics once it gets into the body. And that, from a manufacturing standpoint, that is ideal because the manufacturing footprint then needs to be a lot smaller as well. And then from the delivery platform itself, you see here Genvoy. So as some of you may or may not know, we have an off-the-shelf lipid mix to use for research use. But through the work here that I'm going to present, we are using our own PNI's own proprietary ionizable lipid. Then you'll see here 2 different formulations are used throughout as well. And then finally, what kind of enables all this to happen from a scalability perspective is the nanoassembler manufacturing platform. And I was going into a little bit. It's the microfluidic capabilities that allow the same formulation to be made at bench scale all the way up to clinical scale for GMP manufacturing. And all the work I'm going to present today was not done by me. So in this case, I'm not the expert here. I'm relying on the expertise of our team back in Vancouver. So I really ultimately hope I do them justice and, kind of getting across all the hard work that they had done. But the role of it here is really to be kind of an education tool for everyone listening. So thank you again for being here. All right. So if you're asking yourself, Precision NanoSystems as a technology company, what are they doing making a COVID vaccine? So as I mentioned briefly, so Precision NanoSystems is based out of Vancouver. And through a grant from the Canadian Strategic Innovation Fund called SIF, they were giving money to develop this vaccine for Canada. And then also through that, became where we're currently building now in Vancouver is a full biomanufacturing center to do full-scale GMP manufacturing. But just to kind of overview of the timeline for this project, early 2021, the lead candidates were selected. Also back in 2021, the safety tox study was initiated, and then later in 2021, GMP manufacturing did indeed take place. And then finally this year, the goal is to get these formulations into the clinic. So ultimately, the goal of PNI And as some of you may or may not have heard, we've partnered with sister companies being Aldevron, Paul's, Cytiva, and also VanRx. And ultimately the goal is to really provide clients with the full end-to-end solutions from the mRNA manufacturing itself to the final drug product fill and finish capabilities. So we really want to be able to focus on that and kind of help clients however is needed. But obviously that our kind of expertise here is the nanoassembler technology itself. And then we also have inherent expertise with the downstream processing as well. Because if you think about a lot of the downstream processing techniques I'm going to talk about today, particularly tangential flow filtration, weren't necessarily designed and developed to purify nanoparticles carrying genetic material. So it does require a lot of expertise to do that, and kind of the purpose here, and ultimately kind of instill that knowledge onto people listening here, and people ultimately going through this process of scaling up a formulation. So as I mentioned, the nice thing here is with the nanoassembler platform, the scale up process for the core part of the particle forming process, give you the capabilities and technologies to do that as well. And also, the goal here is to enable, pharma, biopharma companies and CDMOs to be able to go through this process themselves. And ultimately, it's just worth mentioning, all the technology, the equipment technology, can be purchased for use. And also with that, we have expertise to help you be successful too. So with the objective here and what I want to cover, ultimately going from that scale up process, and we're going to go from a 0.2 milligram scale to a 50 milligram scale. And now 50 milligram scale may not sound like a lot, but with self-amplifying RNA, that is the amount that is required for that, or the amount of the batch size that is required for that phase 1 trial. Also go over some of the downstream processing that was developed. Ultimately, the downstream process is going to be tangential flow filtration, some of the criteria and considerations done for going through that. But really kind of overall go through the full process and what the manufacturing is based on a list of characteristics. And these are both formulation activity, stability, scalability, repeatability, and identifying those critical process parameters. So first, just to give an overview of how this is being done from the nanoassembler technology side of things. So we use, through development, we use these three platforms, the Ignite, the Blaze, and the GMP system. And the difference here is going to be the scale at which these this technology can produce your formulations. So if you see on the left there, you see the picture of the microfluidic architecture. So what's nice about this system and this technology is that architecture is maintained throughout this entire platform, which means that whatever you develop on the Ignite, those characteristics will carry over to the Blaze and GMP system. Now on the Ignite is where we do our formulation development, and that has a flow rate of 12 mils per minute. In all these cases, we're doing a mixing ratio of 3:1 aqueous to ethanol phase. And then on the Blaze is where we do most of our process development work, being our TFF, what that downstream process is going to be. And those runs are done in 115 mils per minute, or it can run up to 150 mils per minute, which is also what these runs are done at. And then finally, GMP system. So we're ready to do full GMP manufacturing in your clean room. You can use your GMP system, and that runs up to 200 mils per minute. And then for downstream processing for this, which again I'm going to go over, Ignite was done dialysis, but then when you do the Blaze and GMP system, you're doing TFF. So just the first kind of data to show here. So throughout this scale-up work, 2 lead formulations were chosen to take through the process. And the difference in this case is the formulation, and we call them here LNP1 and LNP2. So what you see, if you look here on the scale, so this shows a graph of different scales going from Ignite, Blaze, GMP. And there's a scale here going from 0.2 mgs, 2.5 mgs on the Blaze to 50 milligrams on the GMP system. And again, the flow rates too. So it shows, ultimately, the point here is to show consistency across the different scales of the platform, where you have similar particle size in the 60 nanometer range, polydispersity less than 0.2, and encapsulation efficiency over 90%. And it's also worth kind of noting here that at this stage, a lot of the saRNA was provided by Robin Shattuck's group from Imperial College of London. So basically kind of put this at the front here and show, as you're going through your development process, you always want to have good ways to characterize your nanoparticles, because you need to be sure that as you do the scale-up work, that those critical quality attributes are being maintained. So obviously we're always looking at things like particle size, polydispersity, encapsulation efficiency, and you'll see there's a lot, but what's really important is to have a good bioactivity assay to give some insight as to whether ultimately the LNP and the payload are doing what they're supposed to be doing from a functionality standpoint. So here in these two cases, so the bioactivity assay that we had developed at PNI uses the LNPs being added to HEK293 cells and then measuring the spike protein with ELISA. So here you can see a dose-dependent response. So as you change the concentration of encapsulated RNA, you get a dose-dependent response. And you see you get a good response for both LNP1 and LNP2, slightly larger activity results with LNP1. But as you'll see as we go through this development, there's a lot of other things you need to consider to decide what that lead formulation is going to be. Because obviously the manufacturing and stability and other things like that need to be a part of that equation, not just the in vitro bioactivity. So we'd also done some in vivo testing with these two lead formulations, LNP1, LNP2. In this case, this is a mice study where a prime of 1 microgram LNPs was given to the mice, and then 4 weeks later, a boost of 1 microgram was done, and then saRNA was taken at 3 weeks and 6 weeks post vaccination. And just some high level, a lot of results came out of this, but just some high level things to present here, just showing the antibody response. So you see in both cases with the graph on the left here, with LNP1 and LNP2, at weeks 3 and week 6, you do get a good antibody response in both of those formulations, and also and then to show a specific response, the neutralizing antibody response, you see a good response in both of those cases as well. So it shows here with in vitro, in this case in vivo, it shows slightly higher activity with LNP1, but ultimately in the end, both LNP1 and LNP2 are effective at producing those neutralizing antibodies for COVID. And then finally, another in vivo study, just to highlight a high level here, was a hamster challenge study where hamsters were vaccinated, with a prime and then a boost 4 weeks later, and then the hamsters were inoculated with COVID, and then saRNA was taken also, and also on day 3 and days 10, the groups were sacrificed and look at various different weights and then histology. So what you see in that study. Apologize, there's a lot going on here, but bear with me here. The graph on the left, it's showing, or the top left, it's showing the body weight change over time. But what we see, so in this case, the lead formulation was using to dose the hamsters with, and 2 different siRNA candidates were used, so candidate 1, candidate 2. So you see in both cases, as compared to the control in the top left, the body weight is maintained. The middle graph in the top, the lung weight versus body weight. So you don't see an increase in lung weight as you do with the control, and also kind of similar with the mice study, you see a good antibody response and a good neutralizing antibody response as well. And then finally, the graphs on the bottom actual viral load was measured, both by doing a nasal wash and by doing extracting the respiratory tissue and looking at that specifically. But in both cases, specifically where you're looking at the cranial and caudal respiratory tissue, you see a very significant reduction in the viral load with the saRNA candidates. But now I think that what's kind of most interesting to me here is to get into talking about the actual, downstream and mRNA, this in this case, saRNA-LNP production process. So just to kind of walk through what that process is. So on the nanosembler, on the microfluidics, you have the aqueous phase containing your saRNA, and then your ethanol phase containing your lipid components. And your lipid components, I mean, it's in this case I didn't mention it, but it's going to be a traditional LNP formulation where you have an ionizable lipid, your helper lipid, your cholesterol, and then your pegylated lipid for stabilization. And they're coming in particle formation through the microfluidics. And again, that process or those mixing characteristics can be maintained at different scales. And then part of that process is doing the inline dilution. Because remember, as you're making these LNPs coming off of the microfluidic cartridge at a 3-to-1 ratio, they're going to be in 25% ethanol. So the first step is going to be to dilute them just to increase the stability of that formulation. And then as you go into down tangential flow filtration, that dilution is also needed to stabilize your particles as they're going to go through shear forces. So I'll get into a little more details about what that tangential flow filtration is, and then finally your sterile filtration and then fill and finish process. And as I mentioned at the beginning, we now have partner companies to provide the equipment and another area of expertise to help with the downstream processing as well. So what is the basic flow chart of what tangential flow filtration? So this is what you see on the right. So ultimately, the goal of tangential flow filtration, it's a couple of things. It's to remove the ethanol out of your formulation, and then it's also do an exchange with whatever your final storage buffer is going to be. So in the case of these LNPs, were the target product profile was to have them stored at -80, so we need to put them in the appropriate cryo buffer. So if you look at the flow chart on the right, so coming off the nanosembler, either Blaze or GMP system, we're doing inline dilution, and then we're diluting them to a point where they can be concentrated on the TFF system. So diluting enough, and typically will go below about 3% ethanol in cases just to, depending on kind of how well the LNPs can withstand the shear forces, and go through a TFF concentration step. And then after the concentration, then do different diafiltration buffer exchanges to again further remove the ethanol, but also put in that final do the diafiltration, what that final storage buffer is going to be, and then final concentration to desired concentration, and sterile filtration, and finally vialing. So if you look at all the different, variables or parameters to look at when developing a TFF system, so a lot of work goes into that. And then when so there's a lot of kind of justification, a lot of explanation that needs to go into as far as why you chose different either cartridges or buffers or things like that. So what I'm going to show is just kind of one example where we looked at shear rate, but really just to kind of, in terms of types of cartridges, let's kind of cut to the chase here and say, we typically work with hollow fibers versus plates for materials. We have used both PS and MPS, but the current cartridges we're using are MPS. And for molecular weight cutoff, we look at a range of 100 to 500. And then surface area is something, obviously, larger surface area for a higher flux, but then you have to worry about the hold-up volumes with higher surface area. Shear rates. So shear rate is determined ultimately by your flow rate. So a higher flow rate, the goal will be to ultimately have the process happen faster. But then again, as you're increasing that flow rate, those shear rates, those shear forces can have a negative impact on your particles as well. And then transmembrane pressure ultimately is going to be the driving force to push the material through your tangential flow filtration membrane. So studies that we had done. So these are the two systems that we had used. So the two scales again on the blades, we did 2.5 milligram scale. So for that, we use the replicating crossflow system. And then at the larger scale, the 50 milligram scale, we use the Cytiva active flux system. So similar process overall, but just different systems for the higher capacity needed. So when we looked at shear rate, so this is an example looking at shear rate over TFF processing time. So at this stage of the game, we had figured out what the initial dilution buffers were going to be. So we did a two-step dilution process. So we did the initial dilution coming off of the cartridge, did a TFF concentration step, did a diafiltration in buffer 1 and a diafiltration in buffer 2, and that buffer 2 is going to be your final cryo buffer. What I wanted to look at was different shear rates, because now this is at the smaller scale, and see what's the effective shear rate on your processing time, because as you scale up your process and are doing larger volumes, you need to have your TFF process that can be done within a reasonable amount of time. So we looked at 4,000, 6,000, and 8,000 inverse seconds. So again, that would have been done by increasing the flow rate through the system. And really what we saw was going from 4,000 to 6,000, we saw a pretty significant decrease in processing time, going from 185 to 125 minutes. And then when going all the way up to 8,000 inverse seconds, there wasn't a huge decrease in time. So what this means is that we chose 6,000 inverse seconds as their shear rate. And again, just to mention, and then so we use a 300 kilodalton molecular weight cutoff, and then a transmembrane pressure of 4. And then throughout, it doesn't show here, but all the critical particle characteristics were maintained at these processing conditions as well. So another kind of important thing to consider as you're going through your development process is sampling at different stages along the way. So at the 2.5 milligram scale, again, this is with formulations LNP1 and LNP2. On the blue is LNP1, and then the greenish blue is LNP2. So if we looked at particle size and then encapsulation efficiency and PDI. So you see LNP1, the particle size is very well maintained throughout. LNP2, the particle size is very well maintained throughout. LNP1, it meets acceptance criteria, but we do start to see a slight increase in size at different steps along the TFF process, and then after filtration. So it was somewhat of a cause for concern, but still within acceptance criteria. But then when we took this same process and did it at a larger scale, what you see is a lot of those small changes really get magnified at fuller, at larger scale processing conditions. So here you see, with LNP1, you see a pretty significant jump in particle size, but LNP2, that this particle size and PDI and everything are maintained throughout. So for this reason, and kind of the nice benefit of taking two formulations through development is that, if one needs some more optimization or downstream process needs more optimization, but obviously in doing any of these cases, you're always up against the element of time. We did move forward with LNP2 as our lead formulation, and that is going for, and since then we have done, so we took that, scaled up manufacturing, used the formulation LNP2, and we did the full scale manufacturing across several different sites and facilities. And what you see is that, using that process, is those particle characteristics are maintained. You do see one slight decrease in size, but that is attributed to a different saRNA lot. And then looking at that cryo-TM, you see what you would expect to see with an LNP, and that's a very dense core. But ultimately, this formulation has been done under full GMP manufacturing conditions, and is currently being tested in a GLP tox study. So just, final kind of couple of things here before I conclude. You see that, so obviously stability has to be a part of it, and seeing, kind of any concerns, formulation, you should see them right away, or I guess as soon as you could with any stability concerns. So you see here with LNP1, you do see that LNP1 is, size and PDI, the graphs at the top. You do see a slight increase in particle size and PDI over time, versus LNP2, the particle size and PDI is consistent over time. A slight increase at -20, but the final storage conditions in this case are going to be -80, so not a concern. And then I didn't get into biopsy. Analytical methods and techniques are a key part of any sort of drug development program. So developing those analytical methods and kind of words advices, do them as quickly as possible, because as you're going through this process and you make any changes, you want to be able to measure to see if any of those process changes or anything are going to have an effect on your product quality. So with that, I'll conclude. I think I'm out of time here. Sorry. So I just wanted to kind of finish with saying, we were able to take 2 candidate formulations all the way up to GMP size or clinical capacity at small scale. 0.2 and 2.5 mg, both LNP1 and LNP2 had comparable particle characteristics. But then when we moved to the larger scale production, LNP2 formulation did much better, and that was selected for further studies, and ultimately is being moved forward towards into the clinic. So like I said, there's currently GMP batch was made. It's currently being evaluated for release, and those tox studies are ongoing. I believe are finishing up. From what I've heard, is getting very favorable results from the GLP tox studies. So with that, I'd like to acknowledge the whole P&I team. Obviously, they're the ones that did all this work, and some of our collaborators being Rob Shattuck for the saRNA, and then Von Perry, University of Strathclyde, for some of the GMP manufacturing workers too. All right, with that, I will stop. And I know there's not much time, but take some questions here.
It remains to be first to thank everyone for attending today. Very much appreciate it. Hope you found it interesting. Also, of course, thank you very much to Jason as well. Great for his presentation. Very interesting, really nice data. And we're very much looking forward to seeing you. There will be another Teatime webinar at the same time in a month's time. And thanks, everyone. And we'll see you next time. Thank you.
Thanks, everybody. Thanks, Richard.